In chapter 17, we jump over to the topic of quantitative genetics. We introduced this in chapter 4. There's a whole bunch of information surrounding this topic, so we're only going to do a little tiny bit of it. Quantitative genetics is really steeped in statistics and calculations because it involves a variety of genes and a variety of phenotypes, and they're not necessarily easy to distinguish or determine. So we're just going to do a little tiny bit, and this is mostly linked to our lab on polygenic inheritance in fingerprint patterns. So in order to understand quantitative genetics, we have to understand the difference between discontinuous and continuous characteristics. So in a discontinuous characteristic, you have some number, usually a small number, say 2 or 3, maybe a few more, but a small number of very distinct phenotype categories. So if we look at this example with these plants, we've got tall plants and we've got short plants. We really don't have anything in between. We might have a third one that's maybe a medium height or even a couple more, but they are very distinguishable. You can put them into very distinct categories. So another way to think about this is someone is colorblind or they are not. They're partially colorblind, they're fully colorblind, or they're not. There's not necessarily a range in between. However, we can also look at characteristics that are continuous. So in a continuous characteristic, we don't see those distinct categories. Instead, what we see is a range of phenotypes. And none of those phenotypes are going to be different enough from any of the others to be able to put them into the distinct categories. So in this example, we have the whole range from dwarf to tall. We've got everything in between. So we've got the shorter ones, we've got the taller ones, and then we've got this range of other traits that fall in between the tall and the short. Now you'll notice this also produces this lovely bell curve. And what this is showing us is that most of the phenotypes occur in this middle range. And then very few of them occur in the extreme edges of the range. And that's very characteristic of a continuous set of traits. Over here on the right, we see sort of an example of how the number of genes that are involved might affect our phenotype. So for example, if we have all recessive, we get no doses of hormone, and this gives one specific phenotype. If we have all dominant or all wild type, we have the full dose of all 6 genes, and so we get another distinct phenotype. But in the middle here, we get this whole range. All of these combinations of traits produce different doses of hormones, and so we get different levels of height. And usually, more genes equals more phenotypes. And that is very characteristic of a quantitative trait. So our quantitative traits are going to vary, and they are often influenced by a variety of alleles at multiple loci. So what this means, multiple loci, means multiple different genes. So we have several different genes on different chromosomes and their associated alleles, all affecting a particular phenotype. So what we get is a lot of different factors, starting with a variety of different genes that are all affecting these quantitative characteristics. Quantitative characteristics are usually polygenic. Polygenic just means many genes. And so that's this case up here, multiple loci, where we have multiple different genes all affecting one characteristic. Very often, these continuous traits are also affected by the environment. So if you think about skin color is a very common example of a polygenic continuous trait. So individuals are born with their set of genes and they're born with their particular skin color. But then you can have outside influences like sun, weather exposure, all of that, either temporarily or permanently change the color of somebody's skin. You might even have nutritional differences that change somebody's skin color. So our phenotypic ranges may overlap. So if we look back here, we might have ranges where these very similar phenotypes are very, very hard to distinguish. Sorry, I'm clicking the wrong thing. Very hard to distinguish. And then because of that, it's really hard to use any sort of standards to analyze differences in individuals because it's really hard to draw a line between phenotypes. So if you look at individuals with a wide range of a particular characteristic, it's going to be very hard to separate them into very distinct groups. So a couple of quick examples here. Coat color is generally thought of as a polygenic trait. There are lots of different ways that coat color is inherited, but overall it's thought to be affected by many different traits, many different genes all working together. So we've got a couple of dogs over here. One of our classmates sent me these pictures of his dogs and of his gecko. This is his gecko there. So I did a quick Google search to see if there are any traits that are polygenic in geckos just for fun. And like literally a super quick Google AI search result said that these colors, jungle and tangerine, are skin colors in gecko that are thought to be the effect of polygenic inheritance or many genes affecting that trait. So there are lots of different traits, particularly in humans, that are quantitative traits that vary continuously. If you think about sort of any of the differences that you can look at and see in the human population, it's probably going to be polygenic. So height is polygenic, skin color, weight, blood pressure. These are all examples of traits that are considered to be polygenic traits. In this image here, you can see an example of that continuous skin color variation from the very, very light to very, very dark skin color, and then pretty much everything in between. Now, I'm not sure if these girls are all the same age. They look like they probably are. We're going to assume that they're close in age. You can also see that height variation. So from the tallest to the shortest ones over here, you get this whole range of height. I love this image on the right. This is actually from a different textbook. It's not from our textbook, but it really clearly shows the effects of quantitative characteristics on skin color. So this is showing just 3 genes. It's actually thought that there are quite a few more genes involved in skin color, but this is already a 64 square Punnett square, so we don't need to look at anything bigger than this because you get the gist of it. If we assume that every allele contributes a little bit of pigment to skin color, so a little bit of melanin to skin color, the additive effect of all of these alleles together is going to give the particular skin color of the individuals. So here we could think of this light one as this individual over here, the lightest skin color that we could have. This is no melanin produced, which is probably a little bit extreme. Even this individual here is going to have some melanin in her skin. But as we add more and more alleles, so as we add these alleles here, any single allele is going to give essentially the same skin color. So one single allele gives the next lightest. Any combination of 2 alleles, and we can see how we get lots of different combinations of 2 alleles here. Any combination of 2 alleles is going to give a different skin color. So we've got all of these in here that provide a different skin color. So a little bit more of that dose from more alleles. And then as we move over here, as we increase our number of alleles producing melanin, we get what would be considered the darkest skin color. So this one in this image, this one over here. So you can see that the additive effect of all of these genes accumulates and produces this range. So we get this additive effect of each of those individual alleles. And it's really hard to distinguish these specific phenotypes. So it's really hard to sort of draw a line in the sand and say, these girls have light skin color, medium skin color, and dark skin color. We could sort of arbitrarily set something, but the more individuals that you have, the harder it's going to be to put them into those distinct categories. So with quantitative traits, the reason that it's hard to put them into categories is that we often get overlap of phenotypes. So as we are moving along our gradient, we're looking at those dwarf plants and our tall plants, and then we saw that image of all the different heights in between. It's sometimes very hard to distinguish with a continuous trait where one genotype ends and one genotype begins, because you often have these overlapping regions where you might have multiple combinations of genes that are affecting individuals in the same way. And so you can't always distinguish the genotype from the phenotype. You might be able to narrow it down a bit, but you can't always distinguish them from one another. And so that's what makes quantitative traits really hard to sort of quantify, to categorize, and to separate. So today in our lab, we're going to be looking at polygenic inheritance. So polygenic inheritance specifically deals with cumulative effects of many genes. And just keep in mind that each one of those genes, so let's say we have those height genes, those height genes, each one of those is acting in their own cross. And so they are all acting along Mendel's rules. So they're all following their own inheritance patterns. In this case, we're just going to kind of assume that this is normal dominance recessive. But if you've got lots of genes involved, you could have lots of different inheritance patterns as well. So they're following Mendel's rules of inheritance, but they're all acting together. And then, of course, we also have environmental factors that could have an even greater additive effect to those phenotypes. This graph from your textbook illustrates the differences for adding in multiple loci. So up here at the top, we've got just one gene locus, or one gene, with two alleles. And you can see we've got those distinct phenotypes. So we've got the homozygous, we've got the heterozygous phenotype, and we've got the homozygous recessive. And so we can put them in their distinct categories. But then when we add in the second gene, or the second allele, so two genes, this would reflect a dihybrid. And you all know from your dihybrid that you get at least 4 phenotypes in a regular dihybrid. But there are different allele combinations. And so this graph here is showing that there are potentially 5 phenotype combinations. And then when we add more loci, it gets bigger and bigger and bigger. And so again, we start to see that normal bell curve. And it gets more and more defined the greater the number of genes that we have. We also start to increase our phenotypic classes, so those categories of phenotypes, as we increase and add more genes. So as we're looking at these quantitative characteristics, we're always going to be kind of looking at the distribution. So that distribution is going to show you the number of different phenotypes. So on the bottom here, we've got our phenotype categories, and on the side, we've got our number of individuals. So this is going to tell you the relative proportions for each phenotype. And again, as long as your sample size is big enough, you're going to see this normal distribution where the greatest number of phenotypes comes in the middle, and then we have less individuals with that phenotype as we move out towards the edges. So this is called a normal distribution. It just means that it's symmetrical, that both sides represent the same distribution. So usually when we talk about quantitative traits, we're going to talk about the mean. So the mean is roughly equivalent to the average. We're going to calculate it as the average. There's a lot of different statistics we could do with quantitative genetics, but we're going to kind of stick to the mean. We could do standard deviation, we could do variance, we could do lots of different calculations. We don't really need to do that in this class. The mean is just going to basically tell you sort of what the average or the middle point of your data is. So these two graphs, these are both normal distributions. But they reflect different averages. So 10-year-old boys have a lower average height than 18-year-old boys. So they're both showing that normal curve. They're just showing differences in this particular population. We could also look at variance. This is not something that we're going to calculate or really even work on in our lab today, but just kind of keep in mind that even when we have that normal shaped curve, the range of data points can vary greatly. So in this center graph, we have the mean. We have the same mean for both of the smaller or shorter graphs, but we have a wider variance. So variance just essentially means the extended range of the different number of phenotypes. So in the center graph here, we have a small range, a small variance, same mean, but the individual phenotypes are sort of all clustered around the middle. As we move out to the second chart, we have a wider variance. It covers a broader range of phenotypes. And then as we move even farther out, we get a broader range even still. So all this is saying is that we have the same mean, but we have individuals that are spread out over farther and farther characteristics. So we get sort of more extremes.